about expanded preimplantation genetic testing. Thank you.
Okay, thank you. Again, the disclosures. This time I'm going to give a little bit of background on genomic prediction. I partnered with two gentlemen, Stephen Shu, who is the Vice President of Research and Graduate Studies at Michigan State University. He's been developing methods for predicting complex traits from the genome. He's actually a physicist, but has been involved with mathematics and machine learning algorithms like the one that you see here, along with Laurent Tellier, who was at BGI in the Cognitive Development Laboratory. And these guys brought me on because of my experience developing platforms for genetic testing in the embryo so that we could begin to consider testing for polygenic disorders, which is what I'm going to talk about today. So I think this audience is familiar with the concept of a monogenic disorder like cystic fibrosis caused by a mutation in a single gene. And this represents about 1% of the population. When we look at polygenic disorders, we're evaluating thousands of loci across the genome, and for example, with type 1 diabetes. But in general, polygenic disorders represent about 15 to 25% of the population. So what we're doing now with monogenic disease testing in the embryo is really the tip of the iceberg, and what we're doing is to expand testing to more common heritable diseases. And it's really only become possible to do this with the availability of large data sets. This was a study published several years ago where they were able to predict the origin, geographical origin of individuals based solely on their DNA. And at the time, this was based on a pretty simple algorithm involving principal component analysis, and the methodologies for analyzing these data have improved significantly since this study, but it was a pretty exciting thing to see that you could actually determine where someone comes from just based on their DNA. And so we've started to think about it in terms of predicting disease using similar approaches. And again, it's based on having available data. For example, the UK Biobank has 500,000 genome-wide data sets with several phenotypes available to train our algorithms on predicting complex phenotypes, including several listed here. And there are several other repositories being developed, including the Million Veterans Program, which has a little bit more diverse population, but again, several phenotypes to train on. And I think we're going to see a significant growth in the data that's produced and available for these types of studies as we move forward. We've really only recently seen a significant increase in publications around polygenic risk score predictions. And again, I think this is going to continue to grow significantly moving forward. And so I think many people are familiar with how we've predicted these types of diseases in the past. You have, for example, clinical predictors here for osteoporosis. The physician can do a fracture risk assessment, but it only explains a small percentage of the variation in the phenotypes. So the variance in the population is only predicted to a certain extent with these types of clinical predictors. Most people are familiar with GWAS studies. This is a one-at-a-time approach, and there has been some success, but not as much as we had hoped, and it's really based on limited types of data analysis. When you're looking for one particular SNP to predict a complex trait, I don't think that we could expect it to explain as much of the variance in the population as we would like. And so it's really, again, development of these machine learning algorithms that now allow us to predict a vast majority of the phenotypes just based on DNA analysis. The interesting thing here is that we can combine testing. So for example, if you have a clinical risk predicted, it improves the ability to use polygenic risk scores to predict disease. And we can also use GWAS data sets as out-of-sample validation for predictors that we generate from these large repositories. Now, obviously, we're probably not going to have the ability to determine clinical risk in an embryo, such as fracture risk, or, for example, cholesterol levels. But when you do combine these, again, they improve the predictive value of the polygenic risk score based on the DNA. And so instead, what we have is the ability to use family history. and we know that having a family history increases your risk, so then these polygenic risk scores become more predictive. The initial algorithm was validated looking at actually human height, and one of the reasons is that it's a relatively common phenotype, so all of the individuals in the UK Biobank have their height measured, and it's a relatively reliable phenotype measurement to utilize. And so essentially this was important to validate, again, the machine learning algorithm. We went into the data a little bit further to look at how well it was able to predict the height of siblings, and you can see if there was a greater than 5 centimeter difference in height that we had 88% accuracy at predicting which of the two siblings was taller or shorter. And the algorithm has been replicated looking at actual disease states. And I think it's important to note that in this title you can see that we're able to now predict polygenic disease with risk equivalent to monogenic disease. And here are several examples from that study where they were able to predict disease states for, again, relatively common disorders. So now the challenge was to actually get accurate genotypes in the embryo. So if we are able to do that, you could do anything you can do in an adult in an embryo. And one of the things that we incorporated into this methodology was the ability to automate the sample prep. And this has been a big challenge in our field. Manual preparation of samples is cumbersome, and it also increases your QC burden. So I think automating the process was helpful. Initially, we had 2 different amplifications, and if you recall, we've talked a little bit about how it's been difficult to get both genotyping and copy number accuracy in parallel. So initially, we did 2 different amplifications. One, which we thought would give us accurate genome-wide genotypes, which is important for monogenic and polygenic disease prediction, where you need genotype information. And then in parallel, a second amplification that gives you accurate copy number to evaluate both whole chromosome aneuploidy and structural rearrangements, particularly associated with translocations. But we went on to actually find a third method that allowed us to do all 4 tests from the same methodology. And we're able to run 96 samples in parallel, and actually this is less expensive than next-gen sequencing. Okay, so I've also, in most of the platforms, all of the platforms I've developed, focused on performance on positive controls. And this might seem like an obvious thing, but it's actually relatively limited when you look at methods that are commercially available. Trying to find performance on positive controls is relatively difficult. For aneuploidy, we used samples with prior karyotyping. For structural rearrangements, we looked at samples with segmental aneuploidy. And then for genotyping performance, we used a cystic fibrosis model. And for polygenic disorders, a hypothyroidism model. And we were actually kind of surprised that using a method primarily meant for genotyping accuracy also gave us accurate copy number for evaluating whole chromosome aneuploidies, and you can see several examples from cell lines, positive controls. And we, with our software, convert these to a Circos plot, which I think is a little bit easier for patients to visualize and understand the data. This is the performance looking at whole chromosome aneuploidy. We did have 3 false positives, so again, it's not perfect, and we acknowledge there are technical artifacts. It's also important to recognize the prevalence of aneuploidy that we have in the control set, and I think this is something that's been overlooked in terms of how tests perform. So for example, if you had a 10% false positive rate and you applied it to a group with a base level of 20% aneuploidy, then a third of your aneuploidy predictions are wrong. So it's a 33% error rate. Whereas if you applied it to a group that had a 60% overall aneuploidy rate, then you would have 1 out of 7 inaccurate. And that is important to look at when we evaluate randomized controlled trials in different age groups, where the prevalence of aneuploidy can be different. We also looked at the ability to detect segmental aneuploidies. In this graph, what we have are several examples of known imbalance of different sizes, 3.5 megabases to 48. And then we looked within the chromosome, either in the region that we know is supposed to have 2 copies, or in the imbalanced region that, for example, could have 1 copy or 3 copies. And here we were able to show the ability to distinguish imbalances down to 3.5 megabases, and I think part of this is because of the density and number of SNPs that we're evaluating in each sample. The other interesting application we previously published on is the ability to use the genotypes in parallel with copy number for cases of risk of inheriting an unbalanced embryo or translocation. You can actually distinguish between a carrier and a normal embryo so that eventually the children born wouldn't have the same issue with infertility. You could potentially select an embryo that's not a carrier of the balanced translocation using genotype information. Okay, so that's the copy number based performance. And then genotyping accuracy, we started out by just asking what is the concordance of the genotype information we obtain from limited quantities compared to large quantities. And so this is the kind of performance you expect when you have large amounts of material like you might get from an adult. And then you can see we have essentially equivalent accuracy when starting with limited quantities like you might have from an embryo. So this is really the important information regarding the ability to do everything we can do in an adult in an embryo biopsy. And another interesting application when you have both copy number and genotyping, as I mentioned before, is the ability to use allele ratio data. And so I just pulled this from the internet, but you can see that when you have a trisomy, and we're looking at allele ratios, instead of having a 50-50 ratio where you have one of each of the heterozygous SNPs, when you have 3 copies, you can see that the ratio shifts from 50-50 to 1/3 and 2/3. And so this is applicable to evaluating trisomy, not using copy number, but actually using the genotypes. And here's what you would expect in a disomy. Again, the 50-50 ratio here. And actually it's applicable to monosomy where you see no heterozygous SNPs located here. So you can, again, use the allele ratio data, for example, to evaluate the performance of copy number in parallel. And I mentioned earlier the 3 false positives. When we look at the allele ratios for those false positives and the true positives, you can actually distinguish the two. So combining genotype and copy number data can actually improve your accuracy. When we think about single gene disorders, we can use a relatively common method of looking at Mendelian inheritance. And so if you have the parents and an existing child genotyped, here you see that this father is known to be a carrier of the mutation. We know that the child also is a carrier. So you can start to look at nearby SNPs, for example, that are informative, where the carrier parent is heterozygous. And then you can determine which allele is inherited from the father and associated with the mutation in the child, and then use that profile to make predictions in the embryo. And when we look at the number of informative SNPs, because again we're looking at 900,000, we typically have about 60 SNPs that can be used for every position in the genome. And when applied to positive controls, limited quantities, we had actually 100% concordance with the expected genotypes using this methodology. When we start to look at polygenic disorders, it's a little different strategy. Here what we do is, for example, use the UK Biobank to train, and here we had 219,000 individuals with known hypothyroidism status, either cases or controls. As I mentioned, you can use out-of-sample validation, so this is a different population, a GWAS data set of 2,000 individuals, and the performance was also as expected with this set of, in this case, 4,945 SNPs. When we applied it to samples that we ran through our protocol, Again, we had good predictive value for cases, distinguishing cases from controls, an overall accuracy of 83%. We have several diseases. This is just a subset, and you can see the prevalence in the population. Again, these are relatively common. This is our ability to predict those diseases, again, to the level that you would expect for Mendelian or monogenic disorders. And several of these diseases actually do have a significant reduction in lifespan associated with them. So it could be useful for patients to have this additional selection tool to prevent these diseases going forward. There's some considerations in applying this clinically. In the IVF population, we know that there's a higher risk of polygenic disorders, which may have an explanation in evolution. And so the problem there may be that there are fewer embryos for selection, which I'll talk about briefly. But in the general population where you use family history, we would expect a greater number of embryos, similar to what you might expect from a single gene disorder, where patients are typically not infertile. And if you just look at a publication, and the number of embryos typically obtained for single gene disorders is between 6 and 7. Genetic counseling is going to be a big factor to incorporate in these cases as well. So here is an example of what you might obtain where we are able to test for both aneuploidy and polygenic risk. You might have 2 embryos predicted to have aneuploidy and 2 predicted to be euploid. And then we can add a test, for example, here showing coronary artery disease risk. And this is actually a pretty easy situation, which you might not always obtain, but here you would have the opportunity to select an embryo with a low risk of the disease. In another situation, both euploid embryos might have high risk. And so this might be a challenging situation for a patient to consider. I changed the example to type 1 diabetes because potentially having this information might be beneficial. Even though you know that the embryo has a high risk, you don't have an opportunity to select an embryo with low risk. The information can be useful, and this is the case for adults as well. When having information, you can make decisions to better manage disease. And so, for example, with diabetes, there's something called the honeymoon phase where your immune system starts destroying the beta cells, but there are a lot of research trials to try and prevent continuation of destruction of beta cells. So if you had knowledge of this high risk early on, you might be able to intervene and prevent full development of the disease. So what I've shown you is the ability to use machine learning algorithms and novel molecular genetic methods to predict common heritable disease risk. It's the first method that can simultaneously evaluate aneuploidy, monogenic disease, polygenic risk, and structural rearrangements. And I think we're going to see that these polygenic risk scores could be a useful tool for embryo selection. The other option is not having any information to begin with. So I do think that having this additional data will be helpful to, in the long term, reduce the incidence of common genetic disease in humans. I often get asked about the ethics, and it's hard not to talk about my personal story because actually I don't think it's that important what my personal opinion is. And I do think it's unethical for a genetic counselor to incorporate their personal beliefs when educating patients on their options. Instead, we can use documents such as this one where it has been stated that it's ethically justified for serious conditions, but also for reproductive liberty to allow genetic testing for adult onset conditions of lesser severity and penetrance. And in this paper, they strongly recommend genetic counseling, which I do think is going to be an important part of implementing the testing going forward. The other interesting note in this paper is that testing was not available for multifactorial diseases, but of course now it is. I also found a paper by Edwards and Shulman, and they, in 1996, predict the ability to test for polygenic disorders and gene editing. CRISPR technology essentially is that now. And so in the future, there may be an opportunity to not have to discard embryos with these mutations or diseases, but to edit them so that you could cure the disease prior to transfer. And we talked about CRISPR, I think, earlier, but obviously there's a lot of work to do on the safety and efficacy, but I do think that there's such a huge potential to cure disease that we need to invest resources into research like this as opposed to things like mosaicism. There's also several national surveys, including one in the US, indicating that 74% of Americans support the use of PGT for serious conditions, and this was actually higher than I expected. But also taken another step in terms of editing out disease. Many people are willing to gene edit their future children to prevent disease. So I think we're going to, again, see a lot more research in this area, developing more and more algorithms to predict polygenic and common disease. More databases are going to be available to expand testing beyond monogenic and into polygenic disorders. And this also applies to testing newborns and adults. It's not just applicable to a selection of embryos. So for example, we know that children with Down syndrome have a higher risk for hypothyroidism disease, so having a test that can predict that disease earlier on would be beneficial for treatment in those situations as well. And with that, I'll acknowledge the people who helped contribute to the study and take any questions. Thank you.
Nathan, this was great. I have a question. When you go to test for a polygenic condition, do you, you have like a reference library where you know which genes go with which polygenic disease. Do you also test the patient to see which one does he or she have?
Yeah, so I think that the initial strategy is to focus on diseases that there's a known family history. So these patients already know they have a risk to begin with.
But do you still test them and the family like we would do for monogenic disease?
Correct. Yeah, the information you obtain from the parental genomes is important to making an accurate prediction in the embryo. So we do test parental DNA in parallel.
Great work.
Thanks. Uh-oh.
Oh, I'll have another go. I hope I get a slightly longer answer this time. Great stuff. I'm just wondering whether somewhere along the line you'd be thinking about looking for markers for embryo viability. So on the assumption the embryos are euploid and not mosaic, then is it something you thought about to look for sometime in the future?
Embryo viability markers in the genome.
Yeah. So is embryo A more likely to develop into a baby than embryo B?
Well, there are some loci that may be helpful in evaluating risk of miscarriage and risk of aneuploidy in the parental genomes, and then you could evaluate those in the embryo as well. So it is something we're looking at.
Aneuploidy aside, you know, other development.
Yeah, like miscarriage risk, for example.
Nathan, I have also a question. In your panel that you presented for expanded preimplantation genetic testing, you also included breast cancer. But in breast cancer, as you know, it can be also monogenic increased risk factor due to founder mutations and other many real mutations. So my question is, if you do this panel, do you test before so for the common BRCA mutations, or do you find them during the PGT testing, and then how are you dealing with the counseling?
Yeah, so it gets a little complicated there. The risk from BRCA1, I think, is 1 to 3% of breast cancer in general, but that is included in the analysis. Thank you.